Hello everyone, and thank you for joining us for today's webinar, The Future of Microbial Manufacturing, New Chemically Defined Medium for Protein and Plasmid Production. Over the next hour, we'll be highlighting recent advances in the development of a one-part chemically defined microbial medium to simplify workflows and achieve high-density cultures, ideal for recombinant protein or plasmid production applications supporting therapeutics, gene therapy, and vaccine advancement. Our experts are going to share recent performance data and examples of how to further improve process productivity and reduce risks. Joining me today are industry experts from Thermo Fisher Scientific, Neil Sengupta, Research and Development Scientist, Jason Brown, Research and Development Engineer, and Grant Martin, Senior Manager, Global Product Management, Gibco Vaccine Media. We'll be holding a live Q&A session following this presentation. Feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we can't get to, we'll follow up on after the event. A couple of housekeeping notes. If you run into any issues, please try refreshing your browser first, and if you need any additional support, submit a question via the Q&A. With that said, I'm going to turn it over to Grant to get us started.
Thank you, and hello everyone. Thank you for joining today. Very excited to introduce to you Bacto CD Supreme, one of our newer microbial media products, as well as some very compelling data that we've generated using the enhanced single-use fermenter. Thermo Fisher at a glance, we are a world leader in servicing science. We have over 90,000 employees geared towards supporting you and your efforts and your projects. But at the heart of things, we are R&D scientists and engineers. So working to advance science every day and bring you products to make, life-saving therapeutics possible. In today's agenda, we'll cover the benefits of animal origin-free chemically defined microbial fermentation media by Neil. Results for AAV and LV production, enhanced single-use fermenter, eSUF system, enhanced single-use fermenter case studies with Bacto CD Supreme, and then we'll wrap things up with a conclusion by myself. So starting with the bioprocessing network, which I mentioned, the Gibco cell culture media is made up of. We also have purification, pharma analytics, production chemicals such as buffers, and single-use technology, which is where the enhanced single-use fermenter can be found. These are all businesses found within the bioproduction group that go to supporting bioprocessing customers as a whole. Recently, and over the past several years, we've made significant investments in the bioprocessing network. More recently with grand openings and expansions at the Grand Island, New York site, but also within the single-use technologies business with a new facility in or outside Nashville, Tennessee. So you can see there items in red highlighted are sites that already existed that have been expanded, and then gray is shown with existing footprints. So moving into more of the product, we really just wanted to provide an overview of microbial media, right? So, fermentation has been used for many decades, but wanted to kind of give it an idea in terms of how we approach the development of Bacto CD Supreme. So starting in roughly the 1950s, we saw LV broth developed, and this historically was an animal origin containing product. So typically from porcine components, so animal components, but also with blends of yeast or soy. As we progressed through the years, we saw customers more focused on removing some of those animal components, especially through the 90s as mad cow disease became more prevalent. And then we saw a lot of the formulations begin to be more yeast and soy-based rather than porcine containing products. And that leads us into 2021, when we actually launched Bacto CD Supreme fermentation production media, which is a first of its kind chemically defined microbial media. So we're very excited to share more there. So first, we really wanted to just provide a high level comparison of traditional microbial media that you may be used to, and then compare that to Bacto CD Supreme. Starting with products and its advantages, we'd like to set the foundation to understand what some of those similarities are or some of the differences are in the use and the makeup of the product. So first and foremost, Bacto CD Supreme is not a supplement, it's a complete media compared to a hydrolysate or a peptone like yeast extract that you may be familiar with. So traditional media is protein derived from yeast, plants, or animal sources, and partially hydrolyzed into peptides, free amino acids, sugars, salts, and other components. This media is typically complex and historically referenced to as undefined. Bacto CD Supreme FPM is a defined proprietary blend of amino acids, vitamins, salts, and sugars developed with Gibco's extensive knowledge of cell culture media and supplements. A main differentiator between Bacto CD Supreme FPM and traditional microbial media is sterilization. So historically, microbial media needed to be autoclaved, whereas Bacto CD Supreme has the ability to be both filter sterilized or autoclaved. So hopefully owing to some of the single-use component usage and transitions that we see from classical stainless steel processes more towards single-use applications. As mentioned, in 2021, we were very proud to introduce Bacto CD Supreme FPM, the industry's first commercially available chemically defined medium specifically for microbial fermentation. Since then, we've seen many of the advantages highlighted here demonstrated in practical applications across the world. First and foremost, starting with consistency. A fully chemically defined media ensures you receive a defined media with the ability to achieve consistent performance. The CD product also ensures you will not be introducing any animal origin components into your process, associated viruses, or complex media. Another key testimonial we continue to hear from our customers is the time savings using the media. The media is a one-part base media requiring only the addition of a carbon source or other supplements depending on your E. coli strain. This means most processes can eliminate 20 to 30 components, saving you time with weighing, blending, and hydrating. At 30-liter scale, it's been reported that 5 hours were saved using the product with the potential to eliminate an entire shift from production for scale-up. In early studies, we've shown the product to, at a minimum, achieve equivalent performance compared to classical fermentation medium, and in many cases exceed these standards. These results may vary depending on your specific E. coli strain, so it's essential to understand the nutrient requirements of your strain and following our recommended guidelines for supplementation found in the user guide. We already touched on the flexibility for sterilization, and moving into scalability, that leads us into scalability for large-scale fermentation runs, which Neil and Jason will go into detail today.
So thanks, Grant, for introducing the product. And now let us look at some of the data related to Bacto CD Supreme. So this data shows lot-to-lot consistency that can be achieved by using Bacto CD Supreme. On your left, you're seeing a plasmid production scenario using a DH5 alpha strain. And as you can see from the plot, all the three lots are pretty equivalent to each other. And moreover, you get consistency when comparing to a peptone-containing media such as terrific broth. Similar results were also obtained using BL21 for recombinant protein production. What you're seeing on the plot here is GFP production across three different lots of Bacto CD Supreme, and all of them were outperforming a classic peptone-containing terrific broth media. So not only Bacto CD provides consistency, it can also, depending on your strain application, can outperform or be equivalent to your peptone-containing media. So going further, like how Bacto CD can further help with your workflow optimization, classical E. coli process, and Grant already mentioned that, can have multiple raw materials, especially if it's being made in-house, such as salts, peptones, trace metals, buffers, etc. And there can be about 7 to 20 individual raw materials which would need procurement, incoming quality control, raw material management, component weighing and blending. So as opposed to this classic approach, Bacto CD provides a huge simplification where now it's reduced to just 2 to 3 components, as Bacto CD is a complete ready-to-use media. All you need is your carbon source, antibiotic as per your application. And in certain scenarios, certain oxotrophic strains would need some amino acids. Further, additional flexibility can be achieved for Bacto CD Supreme using sterile filtration as well as autoclaving as per your process. So next, let's look at some data related to comparison in sterile filtration as well as autoclaving. So here's an example of protein production data, again, GFP production using BL21, where we are comparing Bacto CD Supreme, which was filter sterilized, as well as Bacto CD Supreme, which was autoclaved. So as you can see from the plot, that equivalent growth was achieved, as well as equivalent protein production was achieved in both the scenarios, while the data was much better than the terrific broth control in this scenario. So this is a real showcase of the flexibility of the media, where the customer doesn't have to change their process, and it's amenable to both single-use facilities as well as classic stainless steel processes. And here's another example doing a similar study for the approach, where we are comparing Bacto CD Supreme, which was filter sterilized, as well as autoclaved in a 3-liter bioreactor for a DH5 alpha strain for plasmid production. And again, we can see very equivalent growth profiles in both the scenarios, as well as equivalent plasmid production for both filter sterilized as well as autoclaved media. And in this case as well, this media was outperforming terrific broth control. So now that we have seen some flexibility, how Bacto CD Supreme can be used, let's look at performance and scalability for this media. So the plot on your left, you're seeing BL21 growth results in a 3-liter bioreactor done in a batch mode. So essentially what we did was we let the carbon source run out, and but we were comparing the data. So the light blue here is the terrific broth, and the darker blue is Bacto CD Supreme. And when this was compared for protein production, the plot in the center shows that we got enhanced protein production using Bacto CD Supreme media. So it was scalable from shake flask to at least 3-liter bioreactors. Further example of a scale-up was a 12-liter bioreactor where a fed batch study was done. And here in this plot, what you're seeing is light blue is the Bacto CD Supreme, and the dark blue is a classic peptone-containing media. And in this scenario, a simple feed of magnesium trace metals and glucose was used for the BL21 growth. And what we are also achieving is very good optical densities, about OD of 100 plus using this particular. Additional scale-up data for benchtop reactors is shown here using the DH5 alpha strain producing a simple plasmid. What we are similar to the previous plot, what we have the first figure here compares Bacto CD Supreme, which is in light blue, to the terrific broth control for growth. Again, the study was done in a batch mode, meaning the carbon source was let run out. And what we are seeing here is good production as compared to the peptone control media, and the data was comparable to the shake flask data, showing that the media is scalable to 3-liter bioreactors. Additional work was done in a fed batch mode. On to your right, what we are comparing here in this plot is dark blue is the Bacto CD Supreme in a batch mode where we let the glucose run out, whereas the light blue here is the fed batch mode where we provided a very simple feed with magnesium, thiamine, and glucose in this case study. And you can see we were able to achieve optical densities of about 97 to 100. And when we compare the plasmid production between the batch and the fed batch mode, there was a direct proportionality, and we got like about fourfold plasmid production using the fed batch mode. Additionally, we were able to achieve about 1 gram per liter for a very simple plasmid construct we were evaluating in this study. So it again shows that there is a scalability aspect which can occur from both batch to fed batch mode using Bacto CD Supreme. Additional verification was done for the plasmid quality, and this was done on gel for a qualitative assessment. So what you're seeing in this figure is lane 2 is commercially available PUC19 plasmid, which was the small plasmid of for our studies. And lane 1 is the PUC19, which has been linearized. And the main intent of this plot is, are we seeing equivalent qualitative assessment on the plasmid quality across various conditions? So 4, 5, and 6 are shake flask studies where across different plots. So you can see equivalent quality, which is comparable to the supercoiled plasmid available commercially. And then 8, 9, 10 are the batch bioreactor, where 8 and 9 are comparing filter sterilized and autoclaved version of the media, and we can see we are getting equivalent quality there as well. And more so even in the fed batch study where we got about 1 gram per liter, again, we are getting similar equivalent quality. So this was a good proof that the quality is maintained irrespective of the scale, irrespective of the sterilization method, irrespective of batch or fed batch. Additional studies were done on small scale where we evaluated various gene therapy plasmids which were produced in Bacto CD Supreme. So the plot to your left, what you're seeing here, these the light blue bars are the relative fold increase in various AAV plasmid production. In this scenario, this was AAV8 was tested. And the blue dots are the growth, an increase in growth at the harvest time. So you can see there was some increase in growth across using this media the way this was harvested. But more interestingly, there was further specific, huge increase in specific production. So essentially, there was 1.5 to 4, about 3.7 fold increase in AAV8 plasmid production. And especially for the ITR plasmid, which is a harder plasmid to produce in general. Further assessment was done whether these plasmids are being correctly produced through restriction digest. And what we found was in this plot in the middle is that all the bands of the digested plasmid were aligning with the peptone control, which was LB broth in this scenario. So it's telling us the plasmids are being correctly made as well. So not only you're getting increase in production, you're getting good quality to a peptone control. And further, are these plasmids functional? So we took the plasmids and they were transfected into a HEK line, and we evaluated the transfection efficiency, which is shown using these dots, as well as the virus particles per mil, which is shown by this bar. So we are getting at least equivalent, if not better, production using these plasmids produced in Bacto CD Supreme. So similar exercise was repeated for lentiviral plasmids. And again, interestingly, what we saw that PGOI, which is relatively a harder plasmid to produce, we are seeing increase in specific production about 3.8 folds over a classic peptone media like LB broth, as well as the across the board if you look at the dots, which are OD at harvest, they were increasing as compared to LB plot as well. So not only specific production was increasing, there was a slight increase in growth depending on how you harvest the cells. And again, similarly, a restriction digest was done for comparison whether the plasmids are being made correctly, and the results were comparable to LB versus plasmids produced in Bacto CD Supreme. And next, these plasmids were used to transfect HEK cell lines. And what was compared here is transfection efficiency, which are shown in dots, which is for LB versus Bacto CD Supreme, there was slightly increased in transfection efficiency. And moreover, there was further increase in the overall titers as seen in this blue bar when compared to the pepper control. So now that we have seen the application of the chemically defined Bacto CD Supreme for both protein and plasmid production as a next generation of E. coli media, I will hand over the talk to Jason Brown to talk about advancements of enhanced single-use fermenters and further scale up of this media in the new generation of eSUF. Thanks.
Thanks, Neil. As Neil said, I'm Jason Brown, and I am been working for Thermo Fisher for about 10 years as a design and process development engineer. Before working here, I worked in production, scale-up, and media optimization for improved bacterial expression. Before that, I worked in contract manufacturing of various bacteria and yeast strains. It has been really nice for the past 9 years to not have to perform clean-in-place procedures nor change seals. Today, as Neil said, I'm going to discuss the enhanced single-use fermenter and then use of it with the Gibco's Bacto CD Supreme media. So with the original high-performance single-use fermenter, customers have reported reaching about 240 grams per liter wet cell weight and other various densities with E. coli, and also with Pichia pastoris, 300 grams per liter wet cell weight, using the original single-use fermenter, which has Rushton impellers. But one of our highest cell density customers requested higher cooling capacity as their vessel was about 50 feet from the TCU, and they had seen, when scaling some procedures from 300 liters up to 300, that they reached about 190 OD, and they started seeing the temperature vary, so they end the process early. And then also, our single-use fermenters are designed for 60 standard liters per minute and in the 30-liter vessel, and 600 liters per minute in the 300-liter vessel, though most customers prefer to use about half that, or as low as 1 ppm. Some SUF customers reported back to me that they were achieving 400 millimoles per liter per hour of oxygen uptake rate with their procedures, though some of these customers also requested, can we improve the impellers or something to get better oxygen transfer rates, or even to eliminate oxygen supplementation altogether. So to improve cooling capacity, we added a jacketed door for 15% more surface area and a jacketed bottom for an additional 10% surface area. And to help with the most demanding final hours of culture, we added 10% jacketed area above the 300-liter liquid level. This area above the 300 liters of liquid provides cooling to account for gas holdup volume, and as towards the end of the culture, when we're at 300 liters of liquid, it looks more like 350 liters of liquid. So we were able to enlarge the impellers and make modular injection molded versions so the blades could have parabolic shape, which gives significantly higher oxygen delivery. So to talk about a little bit of the design, we worked through various variations and types of geometries and sizes using computational fluid dynamics to evaluate patterns and liquid bubble velocities for mixing performance, power use, KLA, and power number. The modified parabolic impellers provided similar or higher average velocities with lower power use than those of the Rushton impellers. For actual water tests, we printed 3D versions of various of these and compared them for KLA and power use evaluation. We measured what the power actually was with 5 of these iterations, and then finally we settled on these 3 as pictured here from these 3 CFD models, and used those for actual cultures. And then finally we settled on this version on the right, which, even though it's quite a bit larger, we're able to use the same motor and get the same, well, much more oxygen delivery. For best results in heating, cooling, and oxygen delivery, it is important to align the seams of the film to the baffles and pressure fit the bioprocess container to the vessel before adding liquid. As the bioprocess container inflates, the probes and ports are aligned to the windows, and the film moves to fit around the baffles and contacts fully the jacketed surfaces. The weight of the liquid holds the film in place. Temperature shifts were performed with the same TCU and cooling range from -5 to 55 degrees C. Vessels were alternately tested in triplicate temperature shifts, which showed about 23% improvement in heating times and a 25% improvement in cooling times. Later, culture tests were performed with a thermocirculator and 6-degree supply coolant, and the culture showed that increased jacketed area of the enhanced single-use fermentor reached about a 33% higher cell mass with the same E. coli procedure with no temperature deviations before the final feed ran out. Here we have data from the worst case side-by-side comparison cultures with the Rushton 30-liter to that of the enhanced 30-liter and enhanced 300-liter fermenters. These were started on the same day with the same inoculum, feeds, and media. We saw similar final results reaching about 250 grams per liter wet cell weight in all three vessels. The final feed in the 300-liter was slowed by mistake, but we corrected that within about 4 hours of feed starting. The oxygen use in the enhanced 30-liter and 300 lined up before the feed started, and from that time, the feed was corrected through the end of the culture, again showing scalability. The enhanced parabolic impellers reduced oxygen supplementation by 7 times and reduced anafoam use by about half. We had 4 beta sites, 2 from Europe, 1 from Asia, and 1 from the US, who all stated similar results. All these customers compared their cultures with the Russian version, eS.U.F, to that of the enhanced version, eSUF, and also to stainless steel vessels. They use the same procedure and feed rates in these vessels while allowing the dissolved oxygen control loops to ramp up RPM, air, and oxygen supplementation as needed to maintain dissolved oxygen set point. One of these beta site customers said they saw 4 times less oxygen usage, while the other 3 stated that they saw 6 times less oxygen usage or even less. They said the performance was similar to that of their compared stainless steel vessel. One even said that they had improved their plasmid DNA production by 20% over that of the stainless steel. This may have been because they had filtered the media. Of these 3 sites, I know 3 have now moved the enhanced stuff into their GMP production. Many of our customers had asked if we had done a comparison in the stuff with our Bacto chemically defined Supreme media. So we took our existing chemically defined formula and culture procedure, as in the worst case E. coli comparisons I shared previously, and just switched the media out to Bacto CD Supreme. This saved my team lots of time not having to weigh out components nor bringing the pH up to neutral before inoculation. We used 10 grams per liter glycerol in the Bacto CD Supreme media formulation instead of the 20 grams per liter in our standard procedure. So the feed started a little later than the formulated version. The oxygen usage was really close to the same, and we are happy to report that we had 17% higher yields of cell mass from the Bacto CD Supreme media. And to show scalability of CD Supreme media in the eS.U.F, we took the plasmid production procedure from Neil's team in the 3-liter benchtop fermenters and scaled the procedure to 300 liters and adjusted the feed for higher yields. Again, the CD media formulation saved us hours of time and also not having to neutralize the pH before inoculation. This next slide shows a little bit of the growth profile throughout the culture. And we reached about 290 grams per wet cell weight with the Bacto CD Supreme media and about 70 grams per liter dry cell weight. And where normally we only reach about 240 grams per liter wet cell weight and 50 grams per liter dry cell weight. I'll turn the time over back to Grant.
Thank you, Jason and Neil. You've shared a lot of great content and data today. We started with Bacto CD Supreme FPM, performance and consistency, plasmid quality, AAV and LV plasmid production yields and usage, and the enhanced single-use fermenter improvements and scale-up performance using Bacto CD Supreme FPM. A lot of great content and proof of concept here, and I hope the information was very informative and valuable. We have provided a link there to the landing page for the product, which includes several application notes, more detail on the AAV and LV data, as well as a podcast that was recorded with additional information regarding the products. So without further ado, I think we'll move into the question and answer part of the presentation.
All right, guys, thanks. Great presentation there. We have a lot of questions coming in. If you haven't submitted yours yet, please do. We'll get to as many as we can now, but any we don't get to during our Q&A, our presenters will follow up with you afterwards. But we're going to jump right in here to get to as many of these as possible. First one here is, can I use Bacto CD Supreme fermentation production medium for seed stock generation? Who wants to take that one?
I can take that one. So, hi, this is Nilanjan Gupta. So, yes, you can use Bacto CD Supreme fermentation production medium for seed stock generation. And in fact, our recommended procedure is, especially for larger scales, the seed stock be prepared in Bacto CD Supreme for faster growth profiles.
Okay, great.
Thanks.
Let's see, thank you. Is a carbon source required for use with Bacto CD Supreme fermentation production medium? Carbon source required.
Brad, do you want to take this one, or I can take this again?
Yeah, sure, Neil. If you don't mind, I think it's just asking for our carbon source recommendation.
Yes. So the media is designed to be flexible with both using with a glucose or a glycerol. So carbon source, so that has to be added by the customer based on their process. So yes, you do need to add a carbon source with this medium.
Okay, thank you. Moving on, again, great questions coming in. I'm interested in achieving faster growth. How can I ensure successful transition from LB broth to Bacto CD Supreme fermentation production medium? I think that one might be for you, Neil.
All right. Yeah, I think there were some other comments from the audience as well related to that. So for certain, we have a poster related to this. Certain plasmids can induce some lag phase, interestingly. So the way around that is you can add about 2 to 3 grams per liter glucose to speed up the process. The other observation is that sometimes if you are coming from a peptone's frozen stock, it's good to have the seed culture in the Bacto CD Supreme for faster growth. So those will be the two methods, I would say, again, based on your strain, as well as your plasmid of interest, or is it protein production. Like for BL21, we never see too much lag phase issue. It's pretty fast, on par with peptone media. Certain plasmid production constructs will be slightly slower, but overall densities could be much higher than your typical peptone media. Great questions.
Great.
Thank you.
That is wonderful. A lot of great information here. I have one for Jason here. What is the oxygen uptake rate estimated for culture with the enhanced single-use fermenters?
Yes, so with estimating KLA and OER, if using dissolved oxygen probes, most often you are measuring probe response. So the results can vary. But what we did is we actually monitored the off gas and the inlet gas to the fermenters with the Prima BT from Thermo Fisher Scientific. And from that, we were able to calculate the oxygen uptake rate, or OUR values. And we found that with the Rushton SUF, the original SUF with the Rushton impellers, we reached about 700 millimoles per liter per hour at the end of the culture. But yeah, using max out oxygen supplementation. And then we found that with enhanced SUF, around the same time at the end of the culture, we were reaching about 900 millimoles per liter per hour, but we still had 3 times as much oxygen available. So much higher demanding cultures could be achieved in the single-use fermenter, as much more oxygen was available to supplement if needed.
Okay. All right. Let's see. As presented in the last slide, could you explain why the use of the new culture medium reduces risks during the bioprocess?
Yeah, I can take that one. So overall risk reduction is around the composition of the media being chemically defined and now coming from an animal origin source. So to remove that viral risk or other contamination risk. So we feel that's a considerable step towards reducing your risk in your process.
Okay. Well, thank you so much. Going through the rest of these questions here, guys, unless you want to scroll through them, I think a lot of these are very specific and would probably be best addressed directly. So with that, again, if you have, oh, you know what? Here we go. How about this one? How many cell lines have been tested with this media? I'm curious as some, how many cell lines have been tested with this media? I'm sorry, my screen just blanked out for a second there. Media may or may not have all of the amino acid that a particular cell line may require.
Yeah, I can take that
Auxotrophs.
one. Yeah, auxotrophy. So that's an excellent question. And if you go to the website, we do have detailed out typical auxotrophies which can be observed and how to add that. Simply put, like there are certain strains like DH10 which are delta leucine derivatives. So you need to add 0.5 gram per liter leucine and then you're good to go. And so that does talk to about some other protein derivatives as well. Auxotroph. So that's the main type of two auxotrophies we have tested this across couple of lines like DH1, BL21, DH10, stable 3, NEB stable, and others. Like there's such a diversity. So we have tested this media across multiple lines as well. And some of our posters work, which you saw, which is on that website as well as the data for the lentiviral as AAV viral production. They do use auxotrophic strains with supplementation of appropriate amino acids. So it has that flexibility to work across.
All right, so yeah, I'm not seeing any other ones here that I think are related to everyone here, unless you guys have anything else in here, anything else you want to add. Great presentation, a lot of good information there. As I said earlier, anything that you submitted that wasn't answered directly, the SMEs are going to reach out to you and answer any questions that you have. Otherwise, thanks everyone for joining us. I hope you have a great rest of your day. To our presenters, thank you so much, and we hope to see you all again soon.